Hello, my name is Joyce Zandy, and today I will be talking to you on data visualization with Send data to see what happened at the preclinical phase. I am the Send Explorer product lead at Certara, and I am a biostatistician by training. So by training, I am very interested in study data. I am interested in helping scientists see patterns and trends in their data to understand what happens during the course of the studies that they conduct. The outline of today's webinar will be first, I'm going to tell you about what is SEND, and then we're going to talk about what is the impact of the SEND standardization on the pharmaceutical industry. Next, I'm going to talk about some of the benefits of being able to visualize data with Send Explorer, and then I'm going to give a demonstration of Send Explorer. At the end, we'll have some time for Q&A. So what is SEND? SEND is an acronym that stands for the Standard for Exchange of Non-Clinical Data. Now, this specifies a standard format for presenting non-clinical study data in a consistent and structured format, and that allows electronic data review. So historically, data that was submitted to the agency came in the form of a PDF report that was thousands of pages long, that had raw data listings, that had summary tables, and had written report. And the FDA reviewer prior to send would have to flip back and forth between the report and the tables and the listings to try to understand what was going on in the data, to understand what the story was and what the impact of the treatment on the subjects were during the course of the study. And this was very time consuming. If they wanted to get any kind of visual look at the data, they would actually have to transcribe the data manually into a format that could be plotted, perhaps in Excel or some other format. Now, since SEND has come out, the FDA review is much more streamlined. Raw data are submitted along with the submission in SEND format. So the regulatory agency gets the studies, the PDF report with the tables and listings, but they also get the data electronically. And the electronic data includes information about individual subject findings. Now, with that information and a tool like Send Explorer, the regulatory reviewer can quickly not only look at the data, but investigate trends, drill down on outliers to try to understand what's going on in the studies and make better data-informed decisions. So, non-clinical data that's included in regulatory submissions to the FDA CDER, the Center for Drug Evaluation and Research, must now be in SEND format for all studies and data types that are currently modeled in SEND. And these study types include single dose and repeat dose toxicity studies, as well as carcinogenicity studies, as long as they started on or after December 17th, 2016. And safety pharmacology, cardiovascular, and respiratory studies that started after March the 15th of 2019. And it doesn't matter if the study was GLP or non-GLP, all of those studies need to be submitted to the agency in SEND format. Now, FDA CDER, the Center for Biologics Evaluation and Research, will start requiring data in SEND format for the same study types, but for those studies that initiate after March of 2023. So they aren't currently required, although FDA CDER is accepting those studies in SEND format. It will require them for studies that initiate after March 15th of 2023. So what is the impact of SEND standardization on the industry? Well, there's some things that are viewed favorably and there's some things that are viewed unfavorably. The most unfavorable one is that the costs for producing SEND data sets are passed on to the pharmaceutical organizations. So CROs typically tend to generate these for the studies that they conduct, and they pass the costs on to their sponsors. So that adds to the expense that a sponsor needs to bear. On the good side, however, we've got more efficient regulatory data review. So this really greatly enhances the review process, allows the reviewers to make better data-informed decisions and to do it quicker. The other good news is that the pharmaceutical organizations receive study data from multiple CROs in the same format, because as long as they're following SEND, then those CROs will produce data that is in consistent format, and that can be shared easily with the sponsor. Another positive is the development of control terminology lists. So we've got consistent terminology that we're applying when we're looking at cross-study analysis. This is particularly important so that when we're looking at something in one study, we're using consistent terminology to another study, which allows us to look across the studies in our analysis. Another positive is that it allows us to develop tools that facilitate both within and cross-study data summarization and visualization, like SEND Explorer, because now the data is coming in a standard format. So then these tools can leverage that standardization, and then you develop visualizations that lay on top of that. So what can we expect with data visualization and SEND Explorer? Well, we have interactive visualizations that provide scientific insight. The good news is that SEND Explorer is validated. It's web-based application, and it really allows scientists to delve into the patterns and trends in their study. They can look one study at a time. They can look across multiple studies for the same compound, or they can look across an entire portfolio of programs, perhaps to focus just in on historical control data to see what the underlying patterns are in subjects that haven't received any treatment. SEND Explorer supports scientific curiosity. So scientists by nature are curious. They want to be able to understand relationships in the data, and they want to be able to communicate those. So SEND Explorer allows scientists to interact with their data, to notice those patterns, to investigate relationships, and then be able to share that with other scientists. The input for data for SEND Explorer can be interim or final SEND formatted data sets. So that is very easy for SEND Explorer to read. But in addition, it can also read non-SEND formats. So if we have some static exports from a data collection system that's in a consistent format, connectors can be written that will allow SEND Explorer to read that data. Or for sponsors who have a data collection system on site, SEND Explorer can write connectors that will read directly from the live data collection system according to a scheduled refresh period. So that can happen once a day, it can happen overnight, but that way the data, as the study is progressing, the data can be reviewed shortly thereafter by the scientists. Another thing that was important to us when we were developing SEND Explorer is we offer an easy button link to get to data type specific purpose-built visualizations quickly. So you don't have to do a lot of clicks in order to get to a visualization that allows you to understand the patterns and trends in your data. And because different data types have different requirements, the visualizations are built with that particular data type in mind. So we have line graphs for data that's collected over time, or we have bar charts for data that's non-numeric, or we have a distribution plot to look at historical control distribution of data. So they're all built with a specific data type in mind. In addition, SEND Explorer allows organizations to take advantage of the funds that they're using to pay for non-clinical research. So they're investing in these studies. They need the study to conduct the studies for submissions, but then being able to use that data internally for their own decision making, either for that particular program or to look across their entire portfolio, is very powerful and allows them to take advantage of that investment that they've made. And finally, data visualizations with Send Explorer provide that infrastructure that supports a data-driven culture of analytic excellence. And that's very important for pharmaceutical companies to be able to say, we are using data to drive our decisions, and we're using Send Explorer to, to investigate our data and make good decisions based on that. So quickly, some of the benefits for Send Explorer includes identifying and investigating those data trends. As we said, one study at a time or multiple studies at a time. It allows us to extract data from historical findings and produce reference ranges, or we can see what is the frequency of particular findings in subjects that haven't been treated. We can monitor a study as it progresses. We can communicate findings with other scientists, and then again, we can gain internal value from the investment that we've made in producing Send data sets. So I'm going to move into a Send Explorer demonstration, and Send Explorer has both the single study and the multi-study visualization capability. And I've listed here some of the single study visualizations and the multi-study visualizations that are supported by Send Explorer. And you'll notice that there are a number of multi-study visualizations that also are useful in the single study scenario, but they can be extended to multiple studies at once. I'm going to just go through a number of these, a small number of single study visualizations and multi-study visualizations in the application to give you a taste for what the product does. So I'm switching over to my Send Explorer interface, and when I first log into Send Explorer, I am seeing a dashboard that lists all the studies that have been loaded into Send Explorer. So each one of these rows in my dashboard represents a single Send study, and I just have a small number of studies that I'm looking at right now in my training environment. But each one of these is identified by a study ID. That's what the CRO refers to the study as, and then the sponsor reference ID, which may be the same or may be different. That's the number that the sponsor typically refers to the study as. And you'll notice for each one of these studies, I have some basic metadata like the treatment and the species to help orient me to what these studies are. Now, in addition to the few columns that are here, you'll notice that I have a large field that's devoted to all of the domains that are present in each data set. So these include findings domains like body weight and clinical observations like food and water, laboratory measures, macroscopic and microscopic findings, but it also gives me information about the trial design, the trial summary, the trial sets. All of this is more metadata about the subjects and the groups that comprise the study. So all of that information is available right in my dashboard so I can see what data are present for the study that I've loaded. If I'm trying to find a study, I may need some additional information. So I can click this select columns and I can see a lot more fields that are available to me to include in my dashboard. For example, I may want to include the study title, or I may want to include some key date. So I can type in my filter date, and then it will limit my display to any field that deals with a date, and I can select that as well. Now that I have this additional information, I can use it for filtering. Let's say I just want to see rat studies. Maybe I'm looking at rat studies for a particular treatment. So I can filter it down and find the study that I want. Once I do locate that study of interest, let's remove the filtering. I'll get rid of the filtering with this little reset button. Once I find the study that I'm interested in, I can click on either the study ID or the sponsor reference ID to show the details of that study. And this is giving me all that metadata that we were talking about before. I can add a picture, I can add links to external sources, but basically this page gives me an overview of what the study design is all about. It tells me how the subjects were terminated, how many subjects were in each group, males and females. In addition, on this page, I have a little table that shows me endpoints of interest. And as the scientist is going through the study, they can flag endpoints as being meaningful, maybe showing a dose-response relationship, maybe having some sort of unusual outlier formation, that those can be flagged by the scientist who's reviewing the data, and they can be captured in this little table called endpoints of interest, and then the scientist can use that for subsequent filtering. From my view pull down at the top of the page, I can access some specialty visualizations, or I can look at the raw data and some other visualizations through the findings list or some metadata domains. But what I'd like to do is show you quickly how you can get to a visualization using what we call the easy button. So let's say I'm interested in this study 123. It's a dog study and it's a 4-week with a 4-week recovery. I notice the BW domain. When I mouse over that, it says it's going to open a body weight multi-endpoint line graph. When I open that up, it's going to display a line graph in which the groups are organized by sex and dose by default. Each combination of sex and dose appears as a separate legend item and has a different color associated with it. My males are represented with circles, my females with triangles. The same dose level between males and females is colored the same. My y-axis is set up to accommodate the group mean. If I mouse over any line, that group will highlight in the legend. If I mouse over any dot, it will give me details about that particular dot. Now, right now, I'm just looking at body weight data. My male and female data are overlapping. It may be a little difficult for me to see what's going on. So I can always use some subplot options. Let's subplot it. Instead of having males and females together, we'll make males and females side by side. And now I can see what's going on separately for each of the sexes. Furthermore, I can change how I group things. Instead of having sex and dose as my group by fields, I can just make it dose. So I've got males and females side by side, and then just my dose information is differentiating in the legend. So I can mouse over any of these, or for example, I can click on one of those and emphasize it in my plot. It's also possible for me to add other numeric values to my plot. I can add percent change from baseline, which Send Explorer calculates, or I can add any other numeric endpoint. Let's try this albumin one from the clinical pathology domain. Now I can see that I have side by side all of these plots. Since this is a web-based application, I can change my scaling so this fits a little bit better. And when I get it to something that I'm happy with, maybe I want to communicate this with another scientist, I can click this export to SVG. It will download the image in publication quality SVG and allow me to publish that or put that in my PowerPoint or Word document. And it is scalable, so I can drill way down and it keeps its nice crispness. So whatever configuration I have in the plot, it will repeat in the SVG export. I can also drill down and look at individual subject data from this. So again, this is the group mean. My y-axis is optimized for group mean. But if I want to look at individual subject data, I just have to click the little plus sign next to the group mean. And you'll notice that my y-axis doesn't change until I say I want to rescale the y-axis to include subjects. Now, the y-axis for each one of these endpoints is going to be updated so that I can see all of the individual subjects across any group. No matter what group I click on, the individual subjects will display and it doesn't need to be rescaled. By default, I'm looking at the group mean and the individual subjects, but I can click this, the group mean. I can click it a second time to hide it completely, and now I'm just looking at individual subjects. If I mouse over any particular line, I can see that subject's data across all of the endpoints that I've selected. It also highlights in the legend, I can see that this is subject 331, and I can click on this little arrow here to drill down into what we call a subject view that allows me to see all of the data for this subject, any numeric endpoint that's been collected, any clinical observations that have been collected for this subject, and any histopathology data that's been collected, organ weights, and macroscopic findings. And then furthermore, I can convert these numbers to a graphical display so I can see where my subject is with respect to its and the rest of its group, all the ones that are colored the same, and other groups. So I can follow the trajectory of my subject over time, and I can see where it fell across all of these endpoints. Let's get rid of body weight and make this a little bit more succinct. If I see something that looks interesting, like a dose response trend here between the controls and the high dose group, I can click this little plus sign and capture this as an endpoint of interest that I want to delve into further. If I see a value that appears to be interesting to me, I can click that subject's value. It's going to switch to a new subject. So now you can see I've switched to a new subject, and now I can trace that subject's data over time. So this is what we mean when we allow scientists to investigate their data. All right, if I close that out, I go back to the line graph. At this point, I'm going to go back to my dashboard, and I'm going to take a look at a different visualization. What I'd like to look at now is a clinical observations view. I find a different study because it has more interesting clinical observations. This is a 6-week dog study with a 1-month recovery, and I find the clinical observations abbreviation CL, and it tells me it's going to give me a time course view for clinical observations data. When I click on that, it's going to give me 2 plots, one for males and females, and it's counting the number of subjects in each of the groups at the start of each of these intervals. I can then change this to say I want some particular clinical observations. This is data-driven and listed in order of frequency of occurrence. Let's pick uncoordinated, for example. And I can see during each weekly interval how many subjects from each of the dose groups, 0, 1, 10, and 25, how many subjects had this particular finding, or I can change this to say the number of occurrences during each one of those time intervals. And furthermore, I can say, let's select a bunch of findings. Let's take several that might be of interest to us. And then instead of plotting by sex, I'm going to plot by the finding. So now I can see simultaneously, looking at these findings side by side, what was the occurrence of uncoordinated in my control, low, mid, and high dose? How did that look like over time? What was the trajectory of this during the time course of the study? What happened during the recovery period? And I can see that for uncoordinated, next to tremors, next to repetitive behavior. And what this is showing me is that while uncoordinated started high in the beginning of the study, the repetitive behavior is something that happened towards the end of the study, and I can use that in making decisions about my study. Another visualization I would like to quickly show you, and from the single study point of view, is what we call a severity heat map. And here I'm going to a 1-month tox study in beagles with a 1-month recovery. And actually, I'm going to go to this rat study. I changed my mind. I'm going to go to this rat study. It's a 1-month with a recovery, and I'm going to find the MI domain, microscopic findings domain, and it says when I mouse over it, it's going to open a findings heat map view. Now, microscopic findings are not numeric, but I have various findings and I have severities associated with that. This heat map view shows me all the tissues that were collected in the study. It shows me all the findings that were present in the study and a separate line for each finding and severity combination. So each of the tissues appears as a separate group separated by these gray bars. Vertically, I have the groups going across the top in dose order. Each column represents an individual subject, and then where the tissue and the subject intersect is color-coded based on whether or not that particular subject had an unremarkable finding in that tissue, or if the finding was minimal, mild, moderate, marked, and severe, or whatever the severity scale was that was used in that particular study. So I can readily see, as I look across from control to low to mid to high dose group, I can readily see, was there an increase in either incidence or severity as I go from controls up to the high dose group. And here I see that with hypocellular in the bone marrow femur. If I want to flag that, I can click this little plus sign, and now I've called bone marrow femur a tissue of interest. And I can do that similarly for other tissues that demonstrate a dose response relationship. But I can quickly see what the trends are. I can see which tissues were not examined at all because there's no colors there, and I can readily pick up where there is where there are tissues that show a lot of findings. I can then limit this to only those tissues that I have previously flagged as being of interest. I've done that for a number of tissues here, and I can filter this just for the tissues of interest. Okay, so I'm going to quickly now move to show you some multi-study visualizations just to give you a flavor of what they look like as well. If I go to my visualizations pull down, I can go to something called a scatter plot time course view. When I go in here, it shows what I've most recently been looking at. It pulls that back up for me. So I've previously populated this with 2 studies for the same compound. What I'm looking at here is 2 studies for a compound INDS-002. They're both 28-day studies. One is a rat study and one is a dog. Here are the days on which RDW, the selected endpoint, was collected on day 29, 36, and 57 in the rat, and all the days it was collected on in the dog as well. So by default, I'm getting a box and whiskers plot, but I can change this to show the data points so I can see individual subjects and where they fall with respect to RDW at each of the time points on which it was collected for those subjects. And what's nice, since these study designs are so similar, I have day 29, 36, and 57 in the rat study. I also have 29, 36, and 57 in the dog study. I can choose to align these plots by the actual day, which will put 29 over 29, 36 over 36, and 57 over 57. And then I can see this nice dose response relationship from control, low, mid, high. I'm seeing an increase in RDW. I'm also seeing that in the dog study. I actually lost the high dose group, but I can see that pattern that's continuing in the dog study as well as the rat study. All right, so this is a great visualization for being able to compare across studies for the same program. One last multi-study visualization I'll show you is the historical control values distribution plot. When I first go in here, I'm going to see a blank screen, but I can select a test code, for example, cholesterol. Select the one that I want. It tells me how many control subjects I have cholesterol on. And then it's going to give me a distribution plot. It's going to plot the range of values I've seen for cholesterol into however many intervals I want. I've selected 19 intervals, so it's taken the minimum to the maximum of this particular endpoint, and it's giving me a distribution plot. How many control values for rat were seen in each one of these intervals, so I can tell if it looks like it's normally distributed, if the distribution looks similar between males and females. And this is just using historical control data, so I can use that to put findings from my current study into perspective. Right now I've got rats, I've got dogs, and I've got monkeys that are together in this plot, but I can filter it and then do further breakdown by supplier or by age. There's many different things that I can use in generating this historical control. Okay, so I gave you a smattering of visualizations that I showed you, and now I would be happy to take any questions that you might have as a result of the demonstration.